করবো
না লাইভে ঢুকুক হ্যাঁ ঠিক আছে শুরু করে দেখো যদি রেকর্ডটা দিচ্ছে তোমার ভালো
টাইম তো হয়ে চললো আর কি record on this computer লাইভও এসছে
ঠিক আছে
okay
okay now we are live it's 1 minute to go okay let's start to introduce something thanks everyone for attending our 49 webinar in this series hope those are already registered got my email with the instruction otherwise Shoma will introduce little bit rules of how to collect the certificate we will give the link for certificate collections or feedback after the presentation during the end of this webinar, like during the discussions or interview session. Today, Shoma will help me to introduce the speakers and Dr. Tamin Islam from Dhaka University will help us to interactive session. We are very lucky and to get the chance to present the eminent scientist from the Dhaka University. She is working a long time on abiotic stress and quite reputed in Bangladesh and internationally. So I request Shoma to introduce our guest and all the participants, please comment your questions related to this webinar on YouTube chat box. And we are provide the certificate application or feedback link in the YouTube chat box only. Don't communicate regarding this link on WhatsApp or any other media. So I again welcome all of you. Shoma, please go for the introductions a little bit for Bioingene and speakers.
Who is the view in speaker view? Can you check?
It is speaker view.
Okay. I will start. I welcome all of our viewers to Bioingene, a platform from which plant science researchers and scientists can present their research to the world and future scientists can gain knowledge, perspective, and inspiration. We do this through our webinar series, interview sessions, and publications. Thank you for being a part of today's webinar. As more people are joining in, let me provide some housekeeping information related to today's webinar. Please note that after attending today's talk, you can apply for a certificate of participation. For this, you need to submit the feedback form that will be provided multiple times in the YouTube chat after the presentation. When you fill out the feedback form, please use the same email address that you used in the registration form and remember to mention your full institute name and address. Mismatch in email ID may result in non-identification of participants and your certificate may not be issued. You can collect your participation certificates after 2 to 3 days from our website. Bioingene does not send certificates through email. Please make sure that you have enabled YouTube chat on your device so that you can interact and submit your webinar related questions. We will collect all the relevant questions for our speaker. Our esteemed guest speaker today is Professor Zeba Islam Seraj. Professor Seraj is a Bangladeshi scientist known for her research in developing salt-tolerant rice varieties suitable for growth in the coastal areas of Bangladesh. She is currently a professor at the Department of Biochemistry and Molecular Biology, University of Dhaka. Professor Seraj studied at the University of Dhaka, Bangladesh, obtaining a B.Sc in 1980. She completed her M.Sc from the same university in 1982. She obtained her PhD in biochemistry from the University of Glasgow in 1986 and went to the University of Liverpool for post-doctoral work in the following year. After completing her postdoc, she joined the Department of Biochemistry and Molecular Biology, University of Dhaka in 1988. She became an associate professor in 1991 and later a professor in 1997 at the same university. She has been supervising plant biotechnology projects funded by foreign and local grants as a principal investigator since 1991. She has been a visiting researcher at the University of Texas, Austin since 2013. Professor Siraj has established a well-equipped plant biotechnology laboratory at the University of Dhaka. She has been a co-principal investigator in several projects such as the Generation Challenge Program, an initiative to use molecular biology to help boost agricultural production. Professor Siraj has not only worked on fine mapping of major QTLs for salinity tolerance in Hokali, but also characterized traditional rice land races with the aim of finding genetic loci responsible for salt tolerance and applying markers linked to these loci to aid breeding programs for incorporating of salinity tolerance in rice. She also worked on developing genetically modified rice varieties with improved salt tolerance suitable for growing in the coastal regions of Bangladesh. She was the recipient of the Peer Award for using next generation sequencing technologies to find the basis of salt tolerance of a rice landrace endemic to Bangladesh coast. Professor Seraj has been a visiting scientist in Erie, Philippines, and the USDA research station at Beaumont. She has been honored with the visiting researcher status at the University of Texas from 2014 to 2020. She was awarded the Ananya Award 2017 for her contributions to scientific research. She has published in many prestigious journals and she is also a reviewer for top journals in her field of research. We are highly honored and happy to receive her as our guest speaker for today's webinar on the topic Climate Resilient Rice Using Biodiverse Resources. I now request Professor Saraj to kindly share her screen and begin the talk.
Thank you very much, Shoma, for the nice introduction. Let me share my screen. You can see my screen? Yes, okay. So I'll start my talk. Hello, I am excited to be here. Thank you very much to Shuvo and Shoma and Tahmina for inviting me, and I'm quite excited that there's so many registered participants and hopefully also many others on YouTube. So welcome to you all. So I'm today going to talk on climate resilient rice using biodiverse resources. My slides are not moving. Just a minute. Okay. So as you know, climate change has increased the sea level, and salinity is being in the coastal areas. Salinity is gradually increasing and going upwards towards many coastal areas. So in Bangladesh, as you can see in the slide, so this on this panel, it has the left panel, it is 1973, and on the middle panel it's 2000. So there was roughly an increase of 0.2 million hectares over these years, about 36 years now, and 2000 and 2009. So from 1973 to 2009, 36 years, there has been an increase of 26.7% in salinity in the soil in Bangladesh, and at the same time, the rice which is grown in these areas, particularly in the dry season, this is projected to decline by 15.6%, and at the same time our population is still rising. So unfortunately, rice, we are a rice eating country. Rice is unfortunately a salt sensitive crop, and in two of its developmental stages, which is the seedling stage or the baby stage, say 2 weeks, it's very sensitive to salt. And it again becomes very sensitive to salt at the maturity state, when there's when there's going to be flowering and the setting of grain. It's even more sensitive than the baby stage. And on the right panel, you can actually see uh, rice planted in the coastal area, and the damage to it due to the salinity conditions there. Okay, so what is the answer? How can we deal with, because we cannot afford to lose our rice production. I mean, we actually need to increase it. So we look to nature for what nature has, so nature's resources. So we have traditional varieties which grow locally, and these traditional varieties have adapted to growth in their locality over many years, and they provide an evolving pool of traits. So depending on the locality in which they are growing. So if I talk about the coastal areas in Bangladesh, so the local varieties there, they will obviously have salinity tolerance because they're growing there. And these traditional land races, we call them land races. These are, they have great biodiversity. So we started working with this particular land race, which it's called Horkuch, and it naturally, so the farmers in that region in the southwest of Bangladesh, they actually, you know, attracted our attention to it, and they were very happy with this rice, and they said that it's salt tolerant. So we found it to be naturally growing in those areas in the southwest where it's very saline, and it has adapted to the saline coastal conditions. So the problem with this rice, you may think, okay, so we already have rice which grows in the coastal areas, but the thing is, it's very low yielding, and it doesn't grow in the dry season. It's mainly, it grows in the the wet season, which is monsoon, and traditionally the soil salinity is much lower during the monsoon season. So anyway, it's very low yielding, so we cannot rely on it for food security, but it's salt tolerant. So interestingly, what happens when there's salt when it encounters salt, there's not much loss in its fertility, and not much loss in its yield. So so what we did is we characterized this Horkuch, and we found it to be grouped along with other known salt tolerant land races, particularly from India, like Pokali, and it grouped together. So physiologically, we did find it to be salt tolerant, and then we did some expression studies, and we also compared it to Pokali in the reproductive stage, and we actually, so the blue is Pokali and the red is Horkuch, and the green ones and the purple ones, these are high yielding varieties. So we actually found that the percentage for fertility and the yield, it was quite close to Pokali. And when we did the transcriptional studies, we saw that there were the enzymes which deal with ROS, reactive oxygen species stress. These enzymes were upregulated. So actually in biotic and abiotic stresses, both there's a upregulation, there's a lot of reactive oxygen species which are produced. So we found that Horkuch had during salt stress, a lot of ROS scavengers, as we call them, the enzymes which remove these oxygen radicals. So that was one kind of mechanism, and we also saw that some of the photosynthesis enzymes were upregulated. So before I go more into Horkuch and how we use that resource, I want to compare what a traditional variety or a land race, how it compares to a high yielding variety. So for a traditional variety, it's usually very tall, and it's photoperiod sensitive. That is, it flowers only once per year, and it sets only a few grains. Most of the photosynthate actually goes into the body of the plant, and as you can see on the left panel, you can see a tall plant, and you can see mostly leaves and very little grain. On the other hand, on the right panel, you can see a high yielding variety, and although you can see leaves, most of the photosynthate is actually geared to producing higher yields, and breeders have selected for these high yielding varieties over many years, but while selecting for high yields, they did not select for any kind of tolerance, either biotic or abiotic tolerance. So what happens is that which I can will show in the next slide. That is, so I've compared this horcoach. If you see the red box, very few grains, and then if you see the red box on the right panel, you can see a high yielding variety and you can see lots of grain. But this situation is under normal condition. There's no salt. Just see what happens when there's salt in the system. So if you add salt in the system, you can see on the left that's still horcoach. What happens is there's not much reduction in yield. Whatever you saw in the previous slide, there might be slight loss, but you can usually have some panicles with a lot of grain. But on the other hand, on the right hand side, it's a commercial variety and you can see absolutely no grain set at all. So these high yielding varieties, they respond to fertilizer, they respond to fresh water, and if the conditions are ideal, they'll give you a lot of yield. But the slightest stress will make them not set any grain, and of course you need grains for food security. So then what to do? So how can we, the landrace is not enough, it doesn't set enough grain for food security, but it can grow in salt, and the high yielding, it gives us enough grain for food security, but it cannot tolerate any stress whatsoever. So what can we do? Maybe we can introduce the salinity tolerance genetic loci from the landrace into commercial varieties. So, or in other words, we study the physiological mechanism and look at the molecular basis in these land races and then try to combine the high yield and the tolerance in a single variety. So that's the aim of all of my work. So then there are a lot of molecular tools now available, and I took this from Singh et al. 2020 Planta. So I will talk about the land race. So the land race, say horcoach. Okay, so what we can do is we can do functional and comparative genomics. That will include gene expression analysis. You can have the transcriptome assembly, RNA-seq. You can do genome sequencing. So then you can go two pathways. One is if you see that tolerance versus sensitivity, if there's a candidate gene or a regulatory gene, you can go the biotech pathway for crop improvement. Or if you have the DNA sequence, you can do molecular marker development. You can either do that through QTL mapping, or you can do it through genome-wide association. So if anybody doesn't understand these terms, we can do it in the question and answer session. So from molecular marker development, you can have even expression QTLs. So why are expression QTLs important? They are important because QTLs define a big region in the chromosome where your tolerance loci is present. But then there might be thousands of genes there. So if you combine expression QTLs through RNA, you do RNA sequencing and you superimpose the RNA sequencing results with the genome, then you can find within those genetic loci which are the genes which are contributing to tolerance. So that's an expression QTL. So anyway, once you have that, you can do integrated molecular breeding approach, and you can do marker assisted backcrossing, and then go for a development of an improved variety. And you can even combine the two. The biotechnological approach you can have, if you can either overexpress, I'll do that in the next slide, details of the biotechnological work, and then once you develop an improved variety, you can go for field trial. So in the next slide, I'm explaining some of the molecular tools for enhancing plant tolerance. So A panel on the left side, that's overexpression. So if I can identify a gene which helps a plant become tolerant, then I can overexpress it using a constitutive promoter, or I can use the endogenous promoter as well. Introduce multiple, introduce a copy. So sometimes stress inducible promoters are better than constitutive promoters. So it depends on the effect of that particular gene. So we can overexpress it, the target protein, hoping that the activity of that protein is going to protect the plant against salinity tolerance, against salinity stress. And then we can silence certain genes. Sometimes the overexpression of certain genes can actually cause sensitivity. We will go into that later. So we can silence that target protein, and that will give us tolerance. Or we can use CRISPR to introduce mutations if we know that a certain gene which works better, say even in the, it can be the promoter sequence, if it works better under stress and provides tolerance, we can introduce mutation, or using CRISPR we can silence a gene. So the advantage of CRISPR is you can actually end up with non-GMO crops, but I will answer that question later. So moving on, I've just given a timeline here for the different techniques available to us for crop improvement. So on the here in the crossbreeding, where you can see crossbreeding, that actually takes, so you cross, that's what breeders do. They will cross an elite variety with a commercial variety, and then you have to do selection to get the right combination of genes. And that kind of selection might take 8 to 10 years. And then again you have mutation breeding. Again you will mutate using chemicals, but then you have to do series of selections. That also might take 8 to 10 years. Transgenic breeding, well, introducing a transgene, it doesn't take that long, 4 to 5 years, but then because of the regulatory requirements, that might take another 10 years, sorry, another 5 years. So again, this is 8 to 12 years. And then if we look at CRISPR or CRISPR-Cas genome editing, because we can end up with non-GMO crops, we can actually release a variety much earlier, which is 4 to 6 years. So I took this from Annual Review of Plant Biology, Chen et al., 2019, sorry. So now coming back to my work, so I'm going to talk in 3 parts. One is, use of fluorescent DNA marker assisted breeding. And then I have some transgenic salt, drought, and heat tolerant high yielding rice. And also using plant growth promoting bacteria and fungi, which is actually isolated from some of these land races or some halophytes which are growing in the coastal areas. So I'm going to talk about all these. So just to summarize what I talked about so far. So if we have a land race, we can characterize it. If we have some genes, we can put it into a commercial genotype, and that needn't be rice. If I use transgenic, the importance of transgenic is I don't have any crossing barrier. So I can cross it into any variety. It can be a distant rice or it can be any other, as long as I have a gene, usually a regulatory gene. There's no genetic barrier for transgenic. Or if I want to cross it, as I said, traditional breeding takes a long time. So I have these molecular tools, and I can use, it's called precision breeding using molecular markers. I can do precision breeding and put it back into commercial rice. As far as conventional breeding has been concerned, there are a few varieties which has been released by our breeding institute, Bangladesh Rice Research Institute. However, only one of them, like Biridhan 67 we call them, that's got a little bit of popularity. Other varieties have not been so good. So, but you need many more varieties for the different agroecological areas along the coastal area, even in Bangladesh. So this project for the marker precision breeding is called fluorescent DNA marker assisted breeding for producing highly stress tolerant elite rice varieties. So we use fluorescent markers, and we used a technique called competitive allele specific priming. So what it does is for the 2 SNPs usually, so we map the gene, the salt-tolerant loci in Horkoch, and we found the SNPs SNP specific to Horkoch which were absent in a commercial variety. And then so we have the SNPs, and we design primers or DNA specific to each SNP. So for one the allele A here, we'll have a tail X, and so tail X is just a DNA sequence, okay, specific sequence. And for C we'll have tail Y, and the next step is we'll have a tail X specific fluorophore, and we'll have a tail Y specific fluorophore. So the specific primers actually go into the next stage, the second round of priming. Only the tail X and the tail Y are specific to the SNP. And in the next stage, the primer is not specific, so that makes the whole process a little bit cheaper. But the advantage of this CASP is that, okay, so then this goes and binds there. And you can actually, when you do the RT-PCR of the progeny DNA, you can actually see the colors and in the graph they produce, and you can choose your whether you want allele 1 or allele 2. You can actually choose your progenies very easily. So that's the advantage of CASP. So you can do hundreds of samples in one day. Easily 1,000 of samples you can find which alleles they have after the crossing. So that's as I say, it's called precision breeding. Okay. So this is just a little bit of methodology. So first we did DART for our Horkuch, the population that we generated by crossing Horkuch with another sensitive variety. Then we did DART sequencing, and we got the SNPs. And after we got the SNPs, we compared it to database sequences of Nippon Bare, and then we got the sequences around those SNPs, and then we went and designed primers for CASP, and then of course we did the RT-PCR. So the methodology that we used is first we select for the QTL region in our progenies after crossing, and then we select for outside the QTL, we select regions which are specific to the background which we want, the high yielding background that we want, and then again we select for high yielding background in the other chromosomes. That's called background selection. So foreground selection is the QTL selection of the QTL, and then recombinant selection is on two sides of the QTL, and then we do the background selection. So that's how the progenies are selected for. And this experiment just shows how we did the screening of the population. So we have Horkuch into IR29, we had a population, and we actually had a reciprocal population, and then we had the F1s and the F2s which they segregate, and we analyzed the DNA here, and we did the physiological screening at F3, and then we selected progenies and did the QTL mapping. And we did it actually two stages. We did it at the seedling stage and at the reproductive stage. If you recall, I said that like rice is sensitive at two stages, at seedling, and this was actually published in scientific reports. So this is the QTL mapping for Horkuch, and this paper we've also submitted to scientific reports. It's under review. So we found a lot of QTLs for different, like shoot length, root length, and potassium transporters and grain yield all across Horkuch. But how do we can't use all the markers for our breeding program. So we had done RNA sequencing as well, and we selected through EQTLs, we selected the major regions which gave us salinity tolerance. And then Bangladesh Rice Research Institute helped us with the breeding, and we helped them in selection of the progenies through this CASP fluorescent marker. So this is just a scheme. This slide is very crowded, but so we introduced the QTLs of Horkuch into three commercial lines. One is called Biridhan 63, one is Biridhan 67, and one is Biridhan 74. So this is the scheme just with one, like Biridhan 63. So we have among the progenies from the population, we had donor lines with the target QTLs. So all the three QTLs were not present in the donor, and you can't really cross Horkuch because, as I say, it flowers once a year. So we had these donor lines with two QTLs and another donor line with one of the QTLs, and each was crossed with the commercial line, and then we crossed the F1s and a double cross, and then successively we back crossed with BR63 to get more of the high yielding background, one here, one there, and then once we got the back cross to F1, these were selfed to get like homozygous lines with the three QTLs. So this is how we selected for the different QTLs. One of the QTLs we used was Saltol QTL, which is present in chromosome 1. This is also present in Horkuch. And then we used a root length QTL, which this root length QTL was very prominent in, in the salt tolerant progenies. So we use this. And then, so again, the circle is allele 1 and the triangle is allele 2, and the heterozygotes are square. So we can select for heterozygous and the desired allele. So that's how we did selection. And we had one QTL for potassium, which is important because when the plant is flooded by sodium, you want, genes which can help retain the potassium. So we introduced these three QTLs into the crossing progeny. Okay, and then, so these ones we selected. So these are all like, the circular. So we wanted allele 1 in all of these cases. Okay, so that was selected for, and we also selected the heterozygotes. So this is backcross 1 actually, backcross 1 F1. So then ultimately we had some plants which had all the three QTLs. Some of it was still heterozygous and we still had to work on the background. So this is just a standard look at some of the genotype of some of the progenies that we had. Okay, and if we look closely again, there are 6 lines here. So 6 of the lines had the Saltol QTL, which is actually red, so either the red or the heterozygous. And again, for chromosome 2, I think this is the root length. We had all these 6 lines had the root one, and also for the potassium. So these are some of the selected lines, and again these 6 lines, these had the Saltol QTL, root length QTL, and the KQTL. And the recovery of parent genome at backcross 2 was something like this. So this is probably at backcross 1. So we had a greater recovery at backcross 2. So ultimately, backcross 2 F2, and now these lines are being advanced to homozygous salt tolerant high yielding lines. So to summarize, we got an indigenous traditional rice Horkuch. It was characterized physiologically and it was genotyped by sequencing, and then we identified salt tolerant loci and identified markers which could track those loci during the breeding program. And among the loci, the ones that were more important, we identified them by expression studies, that is EQTLs, and by identifying genes within the loci which can give durable tolerance. And 3 of these loci were integrated into commercial varieties, Biridhan 63, 67 and 74. So these are some of the pictures, the crossing, and how the seeds have set, and then replanting and then we used to collect the DNA and then do the marker analysis at Biochemistry. So this is Bangladesh Rice Research Institute, and then when the plants were ready, we used to collect we did the DNA isolation actually right there in Biri, and then brought it back to Dhaka University for the CASPs analysis. And then we selected lines through screening again. So once we had our backcross 2 lines, F2 and F3 lines, we did the seedling screening again, and here these are the screening of the lines that were selected for each BR 63, 67 and 74. So some of the lines performed very well. And then we did reproductive screening as well at 100 millimolar salt stress, and we selected some lines which performed well or set a higher number of grain. And so ultimately now we have 12 salt tolerant lines selected for with genetic background either from BR 63, 67 and 74, and these are being advanced for homozygosity, and these are expected to be both salt tolerant as well as high yielding. So they are now probably in backcross to F4 stage. There's still some heterozygosity, so maybe we'll go up to F5 and then finally do the selection or see whether these can be released. So when you release a line, it has to be salt tolerant, yes, of course, because we are selecting for salt tolerance, but it has to have good yields as well, and that will depend on how many lines out of these 12 salt tolerants are actually selected for release. But I'm very hopeful. So moving gears and going into another, the second part of my lecture. So as I said, we characterized Horkoch, okay, and Horkoch, it showed that it had certain transporters which helped it in salt tolerance, okay. So we wanted to investigate the role of these ion transporters in sensitivity to salt. Okay, so some of these transporters were found to be upregulated in the traditional salt-tolerant rice landrace Horkoch, and not only in some of the the lines that we developed which had the QTLs, they also showed like upregulation of these two channels. So one of them is called potassium 2-pore channel, TPKA for short, and there's another one which is called the hack-like potassium transporter. So we studied these transporters, we did the transcriptional analysis under different stress, different timings, and so I'm not going into the details of the experiment, but then we made a hypothesis. So the next slides are going to show the hypothesis. My slides are not moving again. What happened? The slides were moving fine. I don't know what happened. Oh, okay. So then, this is just a hypothesis that we have. So what happens when this is a root cell, okay, soil, this is a root cell, and when the sodium chloride, it enters through a lot of so there are no sodium transporters in the plant. They're all potassium transporters, but unfortunately some potassium transporters allow leakage of sodium because the sodium and the potassium structure is similar. So once you have an over, and potassium levels, concentrations in the soils are pretty low. So whenever there's high salinity, sodium will actually leak into the root cell. So once sodium goes high, uh, because of sodium, actually there's hyperpolarization, depolarization in the membrane, and potassium actually leaks out when sodium leaks in. So through these particular channels, and there is an osmotic and ionic imbalance because of this high sodium. Okay, so what happens then is, as I said, there's depolarization of the membrane, and this depolarization initially inactivates this hack transporter, which is actually specific to potassium, or it allows very low leakage of sodium somehow. So then this in the vacuole you have this TPKA, the 2-pore potassium. So when this happens, this protein is activated and it allows the potassium to go out of the cell, and when that happens, there's no further reduction of potassium, and some little of the ionic balance is restored, and the membrane is no longer depolarized, and at that time hack is activated. So we found that TPK is activated as soon as, say, the salt stress within 1 hour, but hack was activated in 24 hours. So this is the hypothesis. And ultimately, so then what we did is we took Horkoch and we designed guide RNA against these two transporters, and then we transformed Horkoch with it, and we saw whether downregulation of these genes made Horkoch sensitive, and that's exactly what happened. So this slide shows the 2-pore potassium channel. It was downregulated, and we could see that when we did this salinity stress assay, so wild type, this is in the presence of 100 millimolar salt or 150 millimolar salt solution, and if you keep the leaves there for 3 or 4 days, the wild type Horkoch, which is tolerant, it remained green, and the transformed Horkoch actually became yellow. So that's a sign of sensitivity, and we did other sensitivity tests as well. And when we downregulate this 2-pore potassium channel, Horkoch actually becomes sensitive. And then, okay, so we had some of these plants. again, so then we, okay, sorry. So this is the second is the hack. So we did the same for the hack gene, which is in the plasma membrane. We designed the guide RNA for downregulating the hack gene, and again we saw that Horkuch became sensitive. Okay, so when we downregulate both the 2,4-K potassium channel and the hack transporter, Horkuch is becoming sensitive. So then we went on to overexpress. So we now know that the 2,4-K channel is very important, so we cloned it into sensitive rice. We cloned it into sensitive rice, and this is the rice that we got after using a constitutive promoter to clone the 2,4-K channel protein. And on the left panel you can see the transgenic plant under 100 millimolar salt, actually 120 millimolar salt for 10 days. This is what the transgenic look like, which is overexpressing the 2,4-K channel. And on the right panel you have the wild type BR28. You can see that it hasn't grown much, and it is showing signs of much more signs of stress than the transgenic line. Okay, so switching gears again. So then Horkuch, we used it to you know, it used it for some to integrate some of its genetic loci into sensitive rice, and then we showed that two of its transporters are very important in salt tolerance, and maybe we can follow up by putting both genes together, you know, in rice, but we might have to use the stress inducible promoters, because as I said, there's a time lag. So the 2,4-K channel, it's upregulated within an hour or so, and the hack one is upregulated in 24 hours. So let's see what we can do with it. So now changing gears again. So there's this uri dhan which grows actually in the coastal areas of India as well, and Bangladesh. We locally call it uri dhan, and it's Oryza coarctata. So it's the only halophyte which can set grains in rice. Set rice-like grains. Very few. I mean, the land you saw in the land race, there are few grains, and in Oryza coarctata there are even fewer, maybe 10 grains per panicle or something like that. But anyway, since it grows in the coastal areas, we are very much interested in. So it grows through its seeds don't germinate, and it grows through rhizomes. Naturally, it grows through rhizomes every year. So we isolated some of the endophytes. So these are bacteria and fungi which live within the plant. So if you make the external tissue sterile and then you crush it up, crush the roots and the rhizomes up, we then isolated some of the bacteria and the fungus which live inside this halophyte, Oryza coarctata. And this is one fungus we found which had a lot of plant growth promoting activities, which included siderophore action. So siderophore is a protein which helps take iron from the soil and into the plant. And then it had zinc solubilization activity, and then it had biological nitrogen fixation ability. It also had phosphate solubilization activity. So these plant growth promoting activities are very important, because you may know that phosphate is very insoluble, and in general, at least in Bangladesh soils are zinc deficient, and of course nitrogen fixation is a very useful property for a plant to have, and siderophore also. So we are characterizing this bacteria. So this is a fungus which we found, and we have some bacteria also, but I'm not showing those results here. So when we applied this fungus, it's actually a strain of Aspergillus niger. So when we apply it without salt, you can actually see the effect of applying the fungus on the rice, how well it grows on the left panel. And on the right panel, we actually gave it saline stress, and you can see that the control plant is weaker than on the left panel, which has no salt, but the plant on the right, it's growing quite well. The roots are quite good. It's almost comparable to the non-stress circumstance. The plant is very healthy. So now we are characterizing it further. and applying this fungus under reproductive stress and see how it helps in you know maintaining yields under salt stress for rice. Okay, so, and the last part of my talk is over my career of like almost 35 years now. I'm going to retire in January of 2023. So transgenic rice that I've developed over the years for saline and dry soils. So these are a lot of publications. So one of this I was saying that this it's called drought and salt tolerance gene, but it's actually a regulatory factor. So whenever there's stress, this transcription factor is upregulated, and it actually stops the plant from going towards yield. Somehow it depresses the yield or the flowering, or the grain formation. So if we downregulate DST, we should get enhanced yield. So we did that work as well, and we overexpress the sodium hydrogen antiporter in the vacuole. So then I have say about 9 or 10 lines which are in the T5 generation, and they're in the background of like popular commercial varieties like Biridhan 28, Biridhan 47, 27, 55, Biridhan 28, and some of them like the G protein. These are drought and heat tolerant. Okay, and then we have rice in which we overexpress the sodium hydrogen antiporter, as I said, so that's salt tolerant, and salt tolerant at 100 millimolar of salt, and it shows certain of course declined yield a little bit, but at least it sets 50% grain yield even after stress. So if it yields say 7 tons per hectare, even after stress is going to set at least 3 tons per hectare. And whereas the traditional varieties or the land races, they don't set more than 1 or 2 tons per hectare. So there's still an advantage with these salt tolerant lines. So I don't know, I'm going to try and release these lines. So they need to be tested further. As I was saying, that the regulatory scenario is very complex. Let's see what happens. So these are some of the lines. So as I said, if I have a non-transgenic under salt stress, it doesn't set any grain. And then we have this the transgenic rice with in the background of BR55, a commercial rice. So that's G protein beta, and this work is actually in collaboration with Dr. Narendra Tuteja. I'll explain about my collaborators further. So this is heat tolerant so this was placed in 45 degrees C for I don't know, a few hours, which is very damaging. And you can see that the wild type is nearly dead, whereas the transgenics are not. And this is with the helicase gene. Here you can see that a lot more grains have set in this. This is actually we transformed a traditional variety. So and then here with the antiporter gene, you can see when we put the complete cDNA with the 5' and the 3' UTR, we get even more seeds set in rice. And then with the same gene, this gene has 3 transcripts. So when we transform it with transcript 1 and transcript 2, again we see quite a lot of tolerance. This is wild type under stress, and these are the transgenic lines under stress. So you can see that they've grown quite a lot under stress, and they yield also better. So these are just some of the lines that I showed you. This is the one with the down-regulated DST gene, and you can actually see, so we made transgenic Kalijira. Kalijira is aromatic rice, and it's prized by many. So you can see that when we down-regulate this DST with artificial microRNA, you get much higher yields. This is not under salt. We didn't do the salt. It's just yield, higher yield. So, and then this is another transporter which is actually down-regulated in Horkuch. It's an ABC transporter. So we down-regulated this gene using CRISPR in IR29 DNA, and we found that by down-regulating this particular transporter, the plant actually becomes tolerant. So this is under 100 millimolar, 120 millimolar stress for a few days. So This is another part. So transgenic rice with key down-regulating genes. So this is my team at the Department of Biochemistry. Some of them are, she's now in UT Austin, almost completed her PhD. She's gone to do her PhD. And this is, he's a collaborator. He collaborates with me, Dr. Rakibul Islam, on the microbial side of my work because he's a microbiologist, but he works in the, he's a professor at the Department of Biochemistry. Shudeep used to be with me for a long time. Now he's completed his PhD. He's doing a postdoc. He's done his work on, he just used guide RNA and down-regulated the starch branching enzymes, and his work was recently published in Plant Journal. And they're all students. This is a PhD student still working. Dr. Sabrina Elias, she was the main, both of these, they worked on the Horkuch. This was a collaborative project between UT Austin and Dhaka University. So she already got her doctorate and she's now an assistant professor at Independent University Bangladesh, a private university, and Taslima is finishing off. She's both of them are wonderful bioinformaticians, and Dr. Ruhul Amin and Dr. Sajadur Rahman are from Bangladesh Rice Research Institute, and they do the breeding while we have been doing the molecular marker studies. And Dr. Sajadur Rahman actually did his PhD under me. So, and then these are some of my collaborators. Dr. Abdel Bagi Ismail, he's from Erie. Dr. Harkamal Wali, Dr. Tom Younger, he's from UT Austin. Oops, sorry, what happened? And Dr. Harkamal Walia, he's in Nebraska. And Dr. Narendra Tuteja, he's at ICGB. I think he's now emeritus scientist at ICGB. And I've received funds from the World Bank, USAID, Peer Science, like World Bank, NATP2, and and of course Bangladesh government. So thank you very much. That's the end of my talk. Thank you for your patience.
Thank you, ma'am. I have collected a few questions, but I will prefer go for the interactive session. So participants get more time and they have the opportunity to comment or any queries on your journey. So I request Dr. Tamina, please go for the interactive session.
Sure.
We have around 300 live viewers. So it would be great to please keep patience. I will give the feedback link just after the interview session. Then we'll go for the interactive question answer round. Over to you, Tamina.
Thank you very much. Thank you, Shubho. Thank you, ma'am, for your nice talk. It is always a pleasure for me to hear you in any kind of webinar or live session here. I'm sure that our audience will be very much happy to know about you and also your research. We already discussed about your research, so I will ask you some questions which will be, like more focused on your personal journey as a plant scientist. Later on, we will move on for some scientific questions that we are collecting from the YouTube and also from our side. So, ma'am, first I would like to know that how you built your scientific career as a plant scientist. Can you please share your journey with us?
Well, it's very interesting. My PhD was on rat liver heteroribonucleoproteins, and that was a long time ago. So this was, like I started work in 1982 at Glasgow University, and then I got my PhD in 1986. And at Liverpool during my postdoc, I actually work on bovine retina. But the journey was very interesting in the fact that when I was doing my PhD, I worked on proteins. And at that time in 1982, recombinant DNA was just becoming popular. But I was too, I couldn't quite catch my PhD problem that I worked on, it would have helped me tremendously if I could use recombinant DNA technology to answer some of the questions that I had. But I had only, the protein technologies at that time to answer my questions. So I was not, if I had gone 2 years later, maybe I could have, because recombinant DNA technique, my supervisor used to go to workshops to learn about recombinant DNA at that time. And when I said that, could I use, and he just said that it's just too expensive, at that time. So then, but then he said, so I moved to Liverpool. Actually, my husband was there. So he said, what I can do for you is I can try and find you a position in Liverpool, but I can't help you with recombinant DNA. But anyway, so when I went to Liverpool, it so happened that was a DNA lab. So, but because I was a protein person, they asked me to do, like in vitro translation of a cDNA library that they had from bovine retina. And that's how I started, and they were very happy with my work and everything. But anyway, then after about 15 months of postdoc, we moved back to Bangladesh, and um, my father is actually, he's a professor of botany. So when I went to Bangladesh, I discovered that doing animal research is very, very expensive. So I talked to my father, and he said, well, biochemistry is biochemistry. It doesn't matter whether you do it in animals or you do it in plants. And you can collaborate with some of my younger colleagues in the Department of Botany. You can use their tissue culture facilities. So then I started work on jute, on the regeneration, on some glyoxylase enzymes, regeneration enzymes in botany department actually. So then once I started working with jute, uh, the Rockefeller Foundation team was visiting. They knew one of our teachers, Professor Anwar Hussain, but Professor Anwar Hussain, he wanted to go for a postdoc at that time. He didn't have the time, and they wanted him to write a grant. So they called me and said that because I was working in plant tissue culture, would I be interested in writing up a grant? So then I applied for Rockefeller, I got it, and then the rest is history. So from Rockefeller, then I started collaborating with Beery, and so on and so forth. And when the NGS technologies came, I got the peer grant, so I could collaborate with UT Austin. So that's how I started, from a protein person to a DNA person, and then, a little bit of, I don't understand a lot of bioinformatics, but enough to be able to supervise projects on bioinformatics. So that's the journey. And the other point that I would like to say that there are no shortcuts to hard work and perseverance. I kept on persisting and persevering, and that, I think perseverance is probably the best key to success. So
Yeah, thank you, ma'am. For your talk or discussion from your journey, one thing I can understand, like what actually motivates you, but it's always fascinating to know, like what exactly that motivates you to become a plant scientist or do something in agriculture. So what is the reason behind? One, already you have mentioned that on that time, animal science was very much difficult or expensive in case of Bangladesh. But I think maybe there are some other reasons for you to work on the plant science or for Bangladeshi agriculture.
okay, so let me slightly answer in a different way. So when I was doing my undergraduate, so I started my undergraduate in 1976. At that time, recombinant DNA was a big thing, and there were lots of companies springing in USA, on based on DNA and recombinant technology. So those were very exciting times. So at that time, that is why I actually chose to study biochemistry. So I've always been excited by recombinant DNA and all of that. But as I said, I did my PhD in proteins, and I mean, a plant cell is more versatile than an animal cell, right? So you can do a lot more manipulations, and it's so easy. Doing tissue culture is easy. You can grow plants easily. We don't even, I mean, we have a very what shall I say, we don't really have an animal house, but we I had a net house, and now we have a greenhouse also. So working for plants is much easier rather than working for animals. So I think, and of course, I mean, plants are pretty to look at. I mean, rice and everything. So, and then very earlier on, I actually had worked with Oryza coarctata a lot, and I had tried to do crossing with Oryza sativa, and part I characterized some of these putative hybrids at Erie because of the salt tolerance thing. And of course, I had collaboration with Erie that also worked out very well. So, because of all these connections, I think that's why I kind of moved on to plant science. And one of my earlier publications on, I started working with jute, and we showed that during regeneration, so jute is a recalcitrant tissue, you can't regenerate it well, you get a lot of chimerics. So at that time I'd shown that when the activity of glyxolase 1 is lowered, that is the time when regeneration occurs. So that work was published in Plant Cell Reports long time ago, 1992 I think. So I mean, early success when I didn't have a big lab, that also led me to think that okay, plant science is quite exciting to do. So that's how I moved forward. And of course, Rockefeller.
Yeah, okay. Like you were talking about the recombinant DNA. Yeah, I'm just having some kind of, like butterflies in my bellies, like just thinking about we just teach in the class so frequently that recombinant DNA is something like that. You got it from one plant or one animal or somewhere else, and you just join it and everything, something like that. At the beginning, you just said that it was quite just new for you also, like, but today we are just moving so fast, and we now have so many new techniques to work on. So my next question will be regarding on that sector. What do you think? Which sector of plant science will, like focus or will be do something good in near future?
Well, food security, obviously. I mean, the prime, particularly for developing countries, I mean, food security, but then not just rice. I think we should, diversify, and we have a lot of our, like grains which are no longer in use. We should, like revive them and work on them too. There are a lot of grains which are naturally salt tolerant. So we should, move in that direction as well. And my main point is that we tend to work in isolation. So we should actually have a multidisciplinary group with nutritionists, social scientists, and say, I know of a professor in Agriculture University at Shere Bangla who has brought in quinoa seeds, right? And he's trying to grow them here. But the thing is, people, I mean, Bangladeshi people or even Indian people, they wouldn't like to eat quinoa, right? So the fact that it's salt tolerant, it's naturally drought tolerant. So you have to combine many things. You might have nutritionists. You can also add like cooks or make tasty dishes out of quinoa to try to make it popular, because we have to biodiversify even with the rice resources. I don't know whether the listeners know that all cultivated varieties, I mean commercial varieties, they have very few genetic origins, like IR8 and a few genetic origins. So that's actually very dangerous. So if one succumbs, all the varieties will succumb to natural disasters or other abiotic stresses and what have you. So we really need to diversify in our food habits. We really need not to just think about ourselves, but to think of the environment and think of what we are doing to it and how to preserve. Because if you even if you, I mean, crop rotation, it's a popular concept, but how much of it is applied? So like participatory collaboration with farmers, that's also very important. So all of that. And I told you I was working with Uri Dhan or Oryza coarctata. I have a climate change B.Sc series, so Bangladesh Climate Change Trust. I have a big grant from there, and I'm doing some field studies. So because Oryza coarctata actually takes up salt water, and it can, by doing so, it reduces the salt water that it's growing in, right? So we are using this Uridhan treated water to irrigate rice in the coastal areas. So we have to look into like nature solutions by nature, to get food security, and it can't be just one. So that's one of the reasons that I also started working with endophytes, because they are also they have plant growth promoting activities. So I think it's a combination of things that will need to combination of disciplines, combination of resources, biological resources, and mutual help with the farmers, particularly in the areas that I mean, I'm living in Dhaka city. What do I know? What's happening in the coastal area? So it has to be, an integrated or a participatory approach to the problems that they face, so that we can answer actually practical problems. So that's the way I think we should go forward.
I completely agree with you about, like about the popularization of new crops. For example, you were talking about quinoa. At my previous lab, like at my postdoc lab, I remember one of my colleagues, she's also a postdoc fellow. She's working on more than 350 lines of different, she collected more than 350 lines of quinoa from the rest of the part of the world. And they are, like doing the field trials to get the popularization. Ma'am, do you think, do you take part any kind of, like social activities or any kind of government or, like legislation part of the thing? Do you take part any of them to, popularization of new crops or introduction of new crops in Bangladesh?
No, I don't. I don't. But these field studies are the first time. So because it's a government project, I visited the field and met with a lot of farmers who were very curious to know I was working with Uridhan, and why I'm being from Dhaka city, why I'm coming to, or why am I interested in their problems. So it was quite amusing, not amusing, I mean, I learned a lot, but it was, their perspective was quite amusing, that they think that the town people, they are not really concerned with their problems. So it was quite a nice feeling, but I don't generally. I mean, it's difficult as it as it is to do research in our university, let alone going out and doing other things. Maybe after I retire, I don't know. Maybe.
And you are talking about that you have a big grant on climate change. So we would like to know some your thoughts on climate change and global warming effects on agriculture in general.
Okay, so I think, because we still need to, as I said, we need engineers. So I'll tell you a story. It's very interesting. So, I've had other grants on the saline coastal areas, right? So this grant I'm talking about was Generation Challenge Program, maybe 10 years or 15 years ago. And at that time, I visited the fields in Shatkira with a plant breeder. And the plant breeder, I mean, okay, he showed me the fields and stuff, and that was it. But 2 years ago, I visited, the areas with an engineer, with a water engineer. And he actually showed me the polders and how the salt water is kept out, and how farmers grow within the polders and outside the polders. And I mean, he explained so many things to me that this was a whole new area that I had never thought of. And because of that, I actually wrote the Climate Change Trust Fund to do this work. So I'm also working in the laboratory, like the endophytes, that's also under the Bangladesh Climate Change Trust. And I'm actually trying to grow uridhan next to rice in a same area, to see the effect that it has in the lab. But then I'm also doing it in the field. But I could only design these experiments because there was an engineer on board. And what I'm trying to say is that we should really look to nature for solutions. As in, when there's monsoon, if we can conserve water at that time, so we need an extensive, and of course the polders also, they need to be reinforced, because there's a lot of leakage of saline water, and salinity is actually increasing quite a lot. So even during my experiment, I faced a lot of problems, as I said, we have this field where we are growing Oryza coarctata. So we give it salt water, say around, I don't know, 150 millimolar, and we keep it there for a month until it comes down to 6. And then we use that 60 millimolar water to irrigate rice. So it's like time sensitive. So so this is boro rice. So normally in those areas they are not able to grow boro rice because they might do so with fresh water, but then they'll lack drinking water. Like fresh water is very scarce during those times. So what happened is we couldn't keep the water, so we had we had sealed, the water was supposed to stay there, it was totally sealed, but all of a sudden the water went down into the soil. So this was in, say, late January or early February. So the soils become so dry that the water that was there, it just went down. So by that time I think the water had reduced from, say, 15 to, I don't know, 9 or something. And then we had to collect water again, and by this time the river water had become much higher, around 20. And then we had to collect fresh water, mix it so that it became 9 decisiemens, and then put it there into the field so that we could irrigate the rice fields at the right time. So these are problems that we don't know about. So conserving water when there's plenty, but then you have to have a system of canals to do it. So here, when I stay in the West, they're already trying to, because of the Western USA, it's becoming so dry, they've already started recirculating toilet water, for not drinking purposes, for other, say, gardening and stuff like that, because they're thinking about the future, like 20 years from now they won't have any water at all. So they're trying to conserve water. But in Bangladesh we are not thinking of these things. We have to think of climate change, we have to conserve water, and we have to activate the local people to thinking of their problems, have talk with them about the solutions of in that locality, and then help them or train them up to realize their own problems, and the government can also help. So it has to be a participatory, each region, like the north in Bangladesh, it has like desertification problems, right? The south has salinity problems, the flooding zones have different problems. So the local people who live there, they know about their problems. So I mean, the agricultural extension workers and the scientists and the government should work together to, under a climate change bureau maybe, in order to plan for the future. That's so important. That's so important for Bangladesh now. So.
Ma'am, let's talk about some problems. You have mentioned that you have faced so many problems, like when you were conducting an experiment or other things, like, for example, to set up a experiment, it's not always necessary that you will get the right results, or you will face some new challenges. So what word of advices do you have for the students or scientists who are working new to the field?
Okay, so one advice I would say that biological experiments, you will have a lot of variability. So you need to start with a lot of replicates, and sometimes when you do your analysis for different like ions, maybe sodium ions or potassium ions or other like photosynthetic pigment, electrolyte leakage, you'll find out that sometimes you always get short of samples, right? So you should start with enough replicates in your experiment, and not only biological replicates, you should do replicates in different seasons as well. Like, be sure of your results before you report anything, because there's a lot of variation, biological variation. So you have to be sure of your results before you report anything. So that's so important. That's so important. People don't realize how important it is, but it is very important, because I'll just give you one example. So we all know that Pokali is very salt tolerant, right? So then what happens is, so there's something called survivability. So Pokali will, say, survive if I give it 100 millimolar salt for, say, 10 days at the seedling stage, at the 2-week-old stage. We'll see that there's a 90% survival, right? But my question is, why do those 10 plants die? Pokali is supposed to be tolerant, right? Why are those 10 plants dying? Nobody has the answer to that. Okay, so maybe it's something like, maybe the seeds are weak, but when you select, you actually select for the best seeds, but you don't know what's happening inside. So now, of course, there's a whole metabolome research. Maybe for those 10 seeds, I don't know, maybe it's a question of timing. Maybe the enzymes are not upregulated. Maybe, the plant was sleepy when the salt stress came and it didn't catch on. I don't know what's it, but so that's what I mean. But there's so much experimental variability. So I do these experiments mostly in a net house. So even if you have a, say, you know, say 1 foot square tray with, a hydroponic system, and you are growing your rice there, one corner of the rice, one corner will be different from the other corner. And of course, we do random sapling and random replications. So, but what I'm trying to say is that even in a tiny area, 1 square foot, if you plant one plant on one side and the other one, there's variability, and when we do the statistics we account for it. But my point is there is variability, so you have to be sure, you have to repeat your experiments enough times that you're actually sure of what you're reporting before you report. So that's so very important.
Okay. Yes, ma'am. Like, I do completely agree with you about the replications and everything. As you mentioned, not only the biological replicates, at the same time, the technical replicates is also very important for our research. Most of the time, what we found that students, when they're working, sometimes they're afraid, like just to report about the failure of the experiment. So we should, like the scientists or the teachers or the group leaders should give the students the confidence to tell every single thing, whatever they are doing. Uh, Talking about this, I would like to ask you.
I would just like I just like to interrupt here. Shuvo is doing such nice work. Maybe they should find start a journal for failed experiments and the reasons why experiments failed. That's not something which is done in the scientific society, but it's so important. It's so important because you learn by your mistakes. So I think there
Yes,
should be
ma'am.
something like that.
Yes, and it's my planning too. Means
Okay.
when and it's just connected. You taught, you just given idea, but we are trying to build some things where we can discuss the failures. So we try to entertain the failures because we learn from the fail, not from the success. So I also believe on that philosophy, and it's a great suggestion, ma'am. Definitely, we will come to you in that way. Thanks. Please continue, Tamina.
Yeah, okay. I have a personal question to you, or it's not really a question, like just out of my curiosity. Like, you have shown so many rice transgenics, like for me, it was like when I did my PhD on rice, it was easier for me to do the transgenics. I worked on Japonica, so it was quite easy. But for indica, I struggled a lot. So not only that, when I came back to Bangladesh, some of my colleagues also, they are like struggling with raising transgenic with indica. So did you publish the protocol that you were using for your
Yes. So because of time constraint, I didn't say. So nowadays we do in planter transformation. Okay. So in planter transformation, we have a method published. I think it's Journal of Plant Biotechnology. But then recently there was another publication on in planter transformation. So we can even most difficult genotypes we are able to transform using in-planter transformation. So in in-planter transformation, you can actually, what you do is you inject and you use your agrobacterium with your construct and you inject into the plumule, and then you do your antibiotic selection, not antibiotic selection, you do just the to kill the agrobacterium, you do that selection and then you just plant the rice. So when those plants flower, so it's like flag leaf dependent, the flag leaves which turn out positive, those panicles, those seeds, those T1 seeds, we again test for positivity. But for the panicles which are not positive, we just discard them. So because we're doing it in the plumule and in the meristematic tissues, not everything is transformed. So it's just a lot at the T1, but it really works. So most of my transgenics are actually using in planter transformation.
Okay.
So you might have seen that in some of my pictures, you saw dark rice. So we use the traditional variety, which was very tissue culture responsive for my earlier work. That was before we started doing in planta. But in planta, before doing in planta, we could only transform a few rice varieties. But after doing in planta, we can transform anything we want to, and
Okay.
it does transform.
That's a very nice argument, because I'm having some grants from government to, like for transgenic in rice. Maybe I will come to you like in
Sure,
person to see.
you're most welcome.
That leads to the next question, actually, ma'am. Can you give us some advice? How can like we can maintain the professional collaboration with the other scientists, not in within the country, but also international collaboration?
Well, the best thing is, I mean, I've talked about this a lot. Nowadays, Hasina Appa, Professor Hasina Khan, she is heading a committee for academic development of Dhaka University. So I actually forgot to mention that to her, but that's a very important point, that there should be a cell in the university which will help with collaboration. So Bangladesh Agriculture University has that BioRes. It has a cell which helps in collaborating, helps in like connecting people. Of course, you will also give suggestions, but sometimes when you're a scientist, when you're teaching, you don't get time, and that particular cell has to be manned by you know scientists who are not maybe I mean bench researchers, but scientists. It can't be manned by people who are not scientists, right? It has to be, I mean, it has to have, say, assistant scientist or whatever, the whole and it has to be manned by like several, there are large fields in biological sciences and other sciences as well. So you should have this cell at the administrative level dealing with the different, and that can also be set according to the priority of the country, what we want in future. So that's so very important, prioritizing research. And after you prioritize research, you can identify the collaborators that you want in your work and then set about doing those collaborations. I'll give you I won't name the country, but I'll give you a story. So this particular country, it was very good in, they had a lot of cotton, okay? So, but they were not very developed, like scientifically. So what they did is they established this institute, and their target was they wanted to mix cotton with, say, elastane to make, say, so that you don't need to iron your cotton, or cotton becomes more versatile and things. So they needed metabolic engineering for that. So they identified, so the laboratory was built, the government put in funds, they took the best PhD students and postdocs and everything. Then the next step is they identified people who were doing metabolic research in advanced countries, and then they sent these people on, like sandwich PhD programs and sandwich postdoctoral programs. They went there, they went and learned, and after they finished, they came back, and the scientists who were supervisors, they were given contracts to come back every year and spend some time so that the laboratory can be set up in the developing country lab as well. So that's how you go about it. You need to know what you want, and then to do that you have to identify what you need to do to get that thing working, and of course you need collaboration with industry as well. So I would suggest that you talk with Professor Hasina Khan, and I don't know why I didn't remember, because I'm going through a personal family crisis right now, so I guess I forget a lot of things. But this is so very important, and I don't know how many times I've repeatedly said that the Dhaka University needs a research cell manned by like scientists who will work out the collaborations, work out the biosafety regulations, and so many things, that we have a commercial unit, so that any of the industries which want their help, they can have that as well. So there's so many things to do which we are not planning enough to do it.
and you are right. Like on 25th, we have the meeting. We were talking about this APD program in our department. Actually, biological faculty organized it in our department, and Professor Hasina Khan was there along with the other members, and we discussed a lot about this. Also about the research cell, we were talking about this biorex thing. We also wanted that in our university as well. Not only this, like collaboration and everything. We also discussed about so many, like things like paperwork that we have to do as a scientist. It's very difficult to maintain your account by yourself and also do the taxes and everything. So we discussed and we raised the question about to maintain a cell that will help us or help the scientists to do that. That is very important for us. I think all over the world we have this thing. I remember my postdoc supervisor, always they have, like one secretary for themselves, like to do the clerical job. But here in Bangladesh, it's quite difficult to have the, I think in India as well, they don't have any secretary for the professors. So you have to do the teachers, lectures, or the research, supervising the students, which make things complicated for us to maintain a good scientific background. Ma'am, now we will talk about the publications. You have so many publications in reputed journals. You have published a lot of papers which are very nice to work with. And at the same time, you also review so many papers and work as peer reviewer for so many journals. So can you give us, or for the students mostly, because for your talk, I think most of the students registered for your talk, can you give some advice to them? How can they really improve their manuscript? While writing the manuscript, not only writing, also after, the pivotal phases, how to handle this?
Okay, so the first thing is, you have to, so you know what you want. Okay, let me go back a few steps. First of all, before you start your experiments, you should think of your paper. We do the experiments and then we think of the paper. That's a wrong approach. So what you should actually do is think of what you would like. You have a general topic of interest, right? So before you start planning the experiments, you think of what journal or what kind of output you want, and then you plan your paper. So what kind of experiments would make your paper acceptable? Then you set out to plan your experiments. That's the best way, but we never do that, right? I've never done it myself, but that's the best way. So the best papers are always planned in advance of even starting the experiment. So that's one advice. And the second advice is you should actually read the general area of, you should read papers thoroughly. Maybe even take as much as a month to read, the good papers. And then try and imagine in your brain, what are the kind of experiments they did and how, what was the story. For every publication, there's always a story. If you can tell a good story, your paper is bound to be accepted. So you have to think of the story. And then once you do that, then you start planning your experiments, and then follow through. And once when you write the paper, be sure you're always working in a group. Everybody doesn't have good writing skills, so you should always make the person who has the best writing skills write the paper, and then somebody can do the literature review, or when you're writing in a group, you have to decide who writes what and everything. It has to be a shared endeavor. And the second thing is that once the reviewer, you have to, I don't like to use the word God, but reviewer is like a God. You have to listen to what he's saying. If you think that they're being unfair, of course, but you always say it politely. Thank you very much for your suggestion, but I politely disagree with you because of this and that. And if you have a good argument and a polite argument, ultimately the editor will have to listen to you, right? Sometimes there are some reviewers who say, oh, this is just the beginning of the experiment, So if it's the beginning of the experiment, I have enough there for one paper. That has to be my next paper. You can't really say that it's, sometimes reviewers do say that. Then, you can politely say, of course, this is the beginning of the story, but this story, it goes from A to say, like T, and the rest has to be in another paper because, of this, and that. But sometimes reviewers, particularly for bioinformatics papers, they say that, okay, your hypothesis is very good, why can't you just do a little bit of experiment to prove it? So if you can do the experiment easily, you should just go ahead and do it, or then you have to move to a lower impact factor for bioinformatics work. So these are the main, I mean, sometimes I have seen writers who do not, they ignore what the reviewers are saying, they ignore it totally and they give it back. So I had this once, I had this very bad experience where, I said that, okay, I have corrected according to the review suggestion, but I forgot to give, only in one place I forgot to give the line numbers, right? Where I had inserted it. And at that time, there was, I mean, email wasn't, they sent me the manuscript back through fax, okay? And it was so rude. They said, if you do not write the line numbers next time, your paper will be rejected outright. And they were so rude. So you have to, when reviewers make a comment, you say you have to bow to them and try to explain as best as you can, to listen to their concerns. So that's very important.
So what
And
I
always,
think of, yes, please
sorry,
continue.
if you're not good in English, once you've written the manuscript, you give it to somebody who may not be in your subject, but who is better in English, because that makes a lot of difference. Good English makes a lot of difference. It's very important. Yeah, sorry.
No, ma'am, it's okay. What like I'm just pointing out or making points that we can, but or the students can take home about the manuscript. Like whatever you mentioned, that planning is very important. First of all, for your paper or your manuscript. Then, secondly, teamwork. That most of the time our students, or I feel that the students do not really care about it. Like the teamwork is very important. Like it's not necessary to you to do all the works, but sharing with the team members that will make manuscript a much more easier to publish or more impactful. And at the same time, I think what you are talking about, the reviews, that's also very important because reviewers always, I don't blame them. It's their work to find out, like, is there any, it's going to be published and everyone is going to read it. So it should be flawless. So they are trying to help us in a way. So take their comments very seriously and point them out very politely so that they can accept in a polite way. So now I will take a privilege, ma'am, to be a woman, a young woman scientist, and to like very famous or reputed scientist to ask a very important question. It will be a gender bias question, maybe, but I will take the privilege to ask you, like, what is your suggestion for the young women scientists in our country or in the world, like, are at their early stages of their career? What should they do, or what will be their focus to do something good in plant science?
Okay, so family support is very important. I mean, if you family support, so if it's not, I mean, if you don't have family support, if you have children, you need somebody reliable at home. If you don't have that, it's very difficult to do good science because you can't have the worry of your family in your mind when you're doing science. So uh, that's very important. So I mean, everybody is not lucky to have family support. So in that case, for Dhaka University, people might laugh, but under the academic research program, particularly for women scientists, you should have a daycare center. You should have a, where you can keep your child, and that daycare center has to be very good. I mean, you have to have highly professional people teaching or, looking after your kids. And, that has to be in every campus so that all teachers and even PhD students can put their children there when they're doing their PhD. That's so very important. If you don't have, support like that, you cannot do research because research is, you have to focus. If you don't focus, you do not find the problems. You can't solve problems. And research is, it's not like a 9 to 5 job where you do mechanical work. It's not that. You have to, think about a problem, think about the solutions, and so on and so forth. And it's very precise science. So you can't do it with if you have problems at home. So I would say for a woman scientist, it's most important to have family support, and The next best is, of course, institutional support, as I'm saying, daycare centers. Otherwise, it's not possible.
Did you face any difficulties, like getting any grant on everything as a women scientist? Like, did you face any kind of difficulties, something like that?
I'm lucky in that I didn't. No.
So then it's a really hopeful message for us, like at least on that part.
Yeah, I mean, you have to write good grants. I mean, you can't, if you write too much that you're going to, like fit in so many different experiments in 1 year, you won't get the grant. So you have to be practical. When you're writing a grant, you have to be practical in what you're writing. So that's important too. I mean, doable. It has to be doable. And you have to show them that you can do it in the time or the money that is being allocated to you. So that's important.
Thank you, ma'am. Shubho, that's from my side. Like, I have finished almost all my queries that I have for Professor Zeba Seraj. Like, do you have anything from your side, from the YouTube questions?
Thank you, Tamina. Ma'am, you covered so many aspects. Sometimes I cannot put these things like what institute should have and what institute to do, but as from your experience and your level of academic positions, you can say it is very nicely. It's not only applicable for Bangladesh, it's also applicable for any developing country. Obviously, we need these things. And here I'm lucky. I have Soma, so my house and my daughter-related tech part is she take care, and it is definitely need a peace of mind to do a good research. And for the collaborations, as you mentioned, generally developed countries have these things, but developing countries are need these things definitely. But we also believe on your ethical point of view, because we also need some ethics to good collaboration. Otherwise, it may be available on department or in university, but it's not working well in that way. So as you can say, Bioingene, it's collaborative. Tamina from Bangladesh, many people are from different countries, and we can do, and we also practice this type of collaboration in that way. Definitely for research, and for nowadays, it's very important for multidisciplinary research, as ma'am mentioned in her talk. So it's a great learning opportunity from you, ma'am. And for the questions related, they are asking, those questions are very basic. So I replied them on instantly, give some uh, Wikipedia link in that way. But I wanted to ask one or two questions, very interesting, like uh, um, someone is asking that these tolerant variety can work anywhere in the world. Means where you produce the salt tolerance rice, is it can be grow in whole world or any specific region?
Well, that's a very interesting question, actually. So,
Yes.
I put them into the background of Bangladeshi high yielding varieties. So, these varieties, these high yielding varieties were bred by Biri and tested in Bangladeshi soil, right? So, if you have climate similar to Bangladesh, I'm sure they will do well. But these are, like these Biri dhans, the commercial backgrounds, these are owned by Bangladesh Rice Research Institute. So I'm sure they can, but then there might be, like regulations and other stuff like that. Because, I mean, being a Bangladeshi, I can use any variety within Bangladesh, right? But I chose because I want them in high yielding background, which will do well in Bangladesh. It should, for climate which is close to Bangladesh, it should work.
Yes, we obviously we hope for the best, and it's sometimes it depend on the environment also. So obviously it is the opportunity that it can be there a high success, but need to know in that way. And uh, one particular questions is related to your experience on jute work. Someone from Bari asking that root development, your comment on vigorous root or root development of jute. So I think it's not exactly related, but still it's from a Bangladesh perspective of questions. Say need to,
I didn't go up to root development because what happened is I was trying transformation in jute. Okay.
Okay.
So I don't know how you transform jute, Shubho, but like transformation. Yeah. So transformation from shoot tip, it ended up in chimeric plants, right? So actually transformation was not successful using tissue culture mediated transformation, and Professor Hasina Khan actually does in-planta transformation of jute. So she was successful in doing in-planta transformation and of course rooting. So I never went up to rooting. So I worked on the enzymes, glycolase, and you know how they correlated with the regeneration ability of the shoot tip in jute. So I never went up to the root.
Ma'am, this is one reason I'm many time I told for my PhD it took 10 years. First 3 years it took for the standardization of media, and I did the transformation on shoot tip transformations. So it's very tough in that time, and yes, I feel these things. And for the olitorias is more tougher than the capsularies, so I choose the capsularies for that. And in 2020 I published another paper for easy transformations. It's an imbibe seed transformation method. It's piercing method, means one sort of implanta but not totally implanta. So it's working and it's very fast in that way. So yes, working on jute is little bit complicated, but the opportunity is the field is open. So there is lots of opportunity to do the work on jute. That I can say it's tough in rice if you go for on that way. So, ma'am, I wanted to limit myself in these two questions. Other questions are easily available on Google or easily available on that way. And we got one suggestion, name of the failure journal of dare to try. So it's quite interesting. Today's participants. I received 309 certificate applications and feedback right now, but it will open more than 5 hours for them. Most of people are rate you 5 out of 5. So all are love it, and there is lot of international scholars and participants are participated. I can see here 22, 23 countries, but maybe more during because it is available in YouTube and anybody can see it in any time. So, ma'am, it's a great pleasure. I thank Tamina to arrange this talk. It's a great, and your story and your suggestions is not only important for us, but it's very important for national policy making process, because our neighboring countries are a little bit similar situations in that way, and it will create a good ecosystem. And definitely if you guys are working on that, and it's very helpful for us, those are the young researchers, and those are the students wanted to come and join plant science research. So ma'am, it's a win-win situation for all of us. Thanks, and for keep your this type of tone to maintain these things. It's very important to something they need push. So yeah, it's a very helpful if you push it, so you can set an example. Already Bangladesh set an example for GM crops, golden rice related work, and BT brinjal. So we are thanks to Bangladesh in that aspect. And it's a great pleasure, ma'am. So we are
Thank
going
you.
to close this session.
Yes. Yeah,
Maybe
thank you
in
very much for
future-
inviting me. Thank you very much. I enjoyed it. Thank you.
Yes, ma'am. It's our pleasure. I will send you all the details within 2-3 days.
Sure. Okay.
Thanks.
Thank you so much.
Thanks, ma'am.
Thank you. Thank you, Tahmina. Thank you, Shoma.
Ma'am, ভালো থাকবেন. Bye.
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 Bye, Tamina.
Bye, Shoma.
Thank you, ma'am.
Bye, Shoma.
Bye. Thank you so much.
It's a pleasure.
Okay. So I'm now ending the